from Microbe TV, this is TWiEVO, This Week in Evolution, episode 42, recorded on April 23rd, 2019. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Greetings again from Elde Lab Studios.
Hey, how was Cold Spring Harbor?
Yeah, it was good. And first of all, I'm sorry that I didn't make it a small detour from JFK to your neighborhood. We were hoping, kind of had penciled in vivo TWiEVO, one of my favorite events in our podcasting pursuits. And unfortunately, just the timing of it got a little crunched. But the meeting at Cold Spring Harbor was really fun. So this was cellular dynamics and modeling. It's been going on for a long time. A lot of cell biology, quantitative and computational cell biology, with a small but growing contingent of folks who are, at the very least, I would say, evolution curious, or evolutionary cell biology as an emerging field. And that was the part of the symposium that I was running about a week and a half or so ago. So Cold Spring Harbor was great, early spring conditions, but didn't quite make it into Manhattan. Next time.
I'm sure I'll run into you this summer sometime.
Oh, yeah. You're kind of all over the map, it sounds like, coming up in the world tour.
Another world tour, yeah. It's good. I like it. I can do it.
Yeah. And then we'll connect for sure at ASV, the American Society for Virology, which is in Minneapolis this year.
If there's a person we could grab for TWiEVO, I could always get a room and we could do it out there too. Yeah.
Or a classic hybrid episode of TWiEVO.
TWiEVO, that's right. We've done that before, right?
We have. Hope to do it again.
Right.
Yeah.
What do we have today, Nels?
Well, we are delving right back into human evolutionary history, the always fascinating case of our own sort of genetic ancestry, our own genetic lineage, the stories tracing back hundreds of thousands of years or even farther in some cases. And so we've done a little bit in this space. We had that fun interview with Sarah Tishkoff, but I think it's been a while since we've kind of considered our own species. And so luckily, there was a really interesting and spicy paper that arrived pretty recently. I think it made a little bit of a splash in the press. And we've actually had a few listeners recommend this paper. Nathan Letts in particular, I wanted to acknowledge, thank you for putting this on our radar and endorsing it as a potential TWiEVO. We love getting these suggestions. I think we'll get a few in our mailbag later. But so the paper showed up in Cell recently. The title of it is Multiple Deeply Divergent Denisovan Ancestries in Papuans. And we'll start to decode all of this in a few minutes. In the meantime, a few of the authors. So co-first authors here. The first is Guy Jacobs. He's at the Complexity Institute. And
That's great. That's great.
I know, isn't it great? This is at Nanyang Technical University in Singapore. And then the other co-author, first author, is probably get the name pronunciation completely wrong, and I apologize in advance, but Georgi Hedjazov, and he is at Massey University. This is in New Zealand. And then also at the Institute for Genomics, I don't know if he moved there recently, in Estonia, the University of Tartu. The lead contact, senior author on the paper, is a fellow called Murray Cox. He's in the statistics and bioinformatics group at Massey University. And I just pulled up his website, professor of computational biology, in a great web address, genomicus.com.
Nice. I like that.
So, yeah. I might be kind of detouring us a little bit, but I wanted to just highlight one of the co-authors who's called Mark Stoneking. He's located at the Max Planck Institute for Evolutionary Anthropology in Leipzig. This is a outfit run by a fellow called Svante Pabo, who's probably familiar to listeners who have had any interest, even a passing or glancing interest, in archaic human genomics. personal connection. So Svante was just here in Utah not too long ago, back in January. And in fact, I think, Vincent, you kind of hinted that we might try for a special episode, which would have been an interview with Svante. I didn't I didn't deliver.
It's funny because you're pretty persistent.
Yeah. Well, and I'll have to confess. So we did have some time together outside of just sort of the strict seminar. And that was, we used it to go skiing up at Elde ski resort.
Is he a good skier?
He's a good skier. Yeah. With a couple of other of my colleagues here. It was a really fun day skiing. And I will say this in my slight defense, at dinner, I did sort of broach the idea of maybe doing a podcast interview in the future. And I have to say that he sounded a little skeptical about he might not be the most sort of podcast interested party, but we'll see. I'll reach out at some point because I think it would be really fascinating to speak with him. And so he was describing for us actually some of the very early forays into doing archaic sequencing. So they had, these bone samples from the Neanderthals, famously, which were some of the first success stories. And so I think there's an early 2000s paper that he was pursuing the mitochondrial genome of Neanderthals. And this was just like a side project or kind of a secret project almost. It wasn't his main sort of thrust at the time, but he was getting really excited about this idea. The paper was published, I think, in Nature. And he now concedes that he basically published his own mitochondrial DNA sequence because it was all contamination. And so, the ideas were all there, the samples and putting it together with the genomic techniques, but it took years of efforts to both identify contamination issues that are rampant for this kind of, sequencing ancient samples. And then, advancing the technology in the field forward with things like clean rooms and all kinds of sort of robust techniques to actually make sure that you're not being fooled by contaminating samples. And the result of that efforts, both by Svante and then others, is, two what are at least currently considered somewhat high quality genomes from 2 Neanderthal samples and 1 Denisovan, which is the topic for today. Going further off the rails here, so I was going to just mention that some of that original work grew out of Alan Wilson's lab, who was at Berkeley, and a really fascinating crew of people. So Mark Stoneking, the co-author on this work, but also great scientists like Mary Claire King, Anna DiRienzo, who's at Chicago now, Rebecca Can, and others. And so this was really kind of a foundational, I think, training ground for molecular evolutionary genetics. So ideas like the molecular clock, building off of genomic diversification, the mitochondrial Eve hypothesis, the emergence of modern humans from Africa. This was Stoneking, Can, and Wilson. Some ideas about how have humans and chimps diverged, and the notion that the coding gene sequences are so nearly identical that there must be other things going on here. This is Mary Claire King, among others, and getting at this idea of the regulatory evolution, that those are the changes. It's not, the genes themselves, but when and where you turn them
98%
on.
98% identity, right?
Yeah, exactly.
But that's the exome, right?
Yeah, exactly right. And so that's, that disconnect was what I think kind of spurred what is now continues to be a fertile ground for research. So really spectacularly interesting lab with great folks emerging from there, which we'll see an example of that, at least peripherally, with Stoneking's contribution to this paper. Kind of a sad story in the sense that Alan Wilson died really young. He's about 56 when he passed away of complications from leukemia, and this was in the early 90s. But I can't help but think that, if he had continued his career along the same way that some of the authors that you and I have spoken to about recent science history, so Carl Woese or Jonathan Wiener and all of the stuff going on, I think Alan Wilson's scientific story would be a really interesting, and the kind of scientists around that would be another really interesting topic.
Yeah, for sure.
Yeah.
There are 2 institutes or centers on this paper I wanted to mention that look interesting. First, the Santa Fe Institute. Do you know anything about that, Nels?
Yeah, just a little bit. I've never been there. Have you?
No, but I'd like to be there. I like sound effect.
Yeah, I hear that. Kind of a posh, interesting kind of desert retreat almost.
Yeah, but there are too many New Yorkers there now. That's the problem.
I see. Fair enough. I mean, I've heard people give talks who've spent sort of time there or done appointments there. A lot of cross-pollination of between disciplines is my sense, kind of big ideas.
It's a nonprofit theoretical research institute.
Mm-hmm.
So I guess they don't do any wet experiments. So.
Yeah, kind of discussing things and colliding ideas in new ways.
And,
yeah.
And the other one is this Stockholm Resilience Center. Doesn't that sound interesting?
It does. I'm not familiar with that one either.
Stockholm.
That's right.
I'm going to look it up. Resilience. Oh, look at there's stockholmresilience.org. And let's see what they do. A reference point for research on global sustainability. Wow. So they do. It's a research institute.
Yeah. It'd be fun to get the backstory on how kind of all these authors contributed to this study.
They do a lot of different things.
Mm-hmm.
Oh, curious. Resilience. Very interesting, isn't it?
Yeah, agree.
This is a big mix of different people from different places all over. We got also University of Indonesia, France, Italy, UK, Indonesia again. Wow, it's just all over the place, right?
Yeah, and I think we'll see some obvious connections given the samples that are analyzed. But yeah, it'd be fun to hear how, and I mean, I guess an illustration too of how for pretty complex studies like this involving pretty heavy lifting with statistical approaches to genomic analysis that you sort of bring together people with all kinds of interesting backgrounds and interesting locations.
Now, Nels, when they talk about archaic homonyms, what do they exactly mean?
Yeah, so we're talking about, so we would be sort of the modern humans, right? Samples, modern humans. That's right. And so
it's Homo sapiens, just right.
Homo sapiens, correct. And then so what led up to this? And if you trace way back millions of years, based on archaeological records of fossil finds, mostly Homo erectus, right? So about from 2 million to a million years ago, and then some diversification. And looks like you found. I'm just looking at the show notes for a minute, and it looks like you found some nice diagrams of this, actually, that you've included.
Yeah, so Homo heidelbergensis, which is kind of contemporaneous with the Homo erectus species, looks like a direct ancestor of both Homo sapiens, Neanderthals, and Denisovans.
Yeah, so kind of a major branch point out of Africa, and with heidelbergensis, then diverging to Neanderthals on the one hand and Denisovans on the other. And so I think, the Neanderthal has obviously been not only in sort of the scientific view for a while, but also sort of, for even just sort of society view, just the idea of what a Neanderthal is, this sort of stereotyped even idea of sort of a primitive human. Less attention has been paid to the Denisovans, and that's actually where this paper will be plumbing in today. Maybe the irony of this is actually instead of sequencing any archaic humans, this is actually the analysis or the new data here involves sequencing of 161 modern genomes from individuals on 14 islands in Southeast Asia, in and around Indonesia. So I think you'd call this island Southeast Asia, and then going even farther east to New Guinea, and with especially a focus on the Papuans of New Guinea.
So we don't have a lot of ancient species or fossils to sequence. So we look in modern humans to see if we can get some information from that, right?
That's exactly right. And so here, the archaic genomes that will be considered, which have already been reported on, are 2 Neanderthals and 1 Denisovan. And the Denisovan is from the Denisova cave, which I believe is in Russia, kind of Siberia. So pretty far north of where the sampling action is for this paper. But as the authors point out, kind of later on, it's actually really hard to get archaic samples or bone or fossil samples in Southeast Asia. It's so humid that there's just not good preservation of DNA in any of the fossil remains that you find.
Yeah, and that cave, of course, is cold, so those are pretty well preserved.
That's right. And so I think that's actually kind of some of the, the advance of this paper is taking the increasing collection of comparative data that's possible by not only having those two Neanderthals, but having that Denisovan, and then connecting that in a new way by doing the sampling in a very underappreciated part of the world when it comes to human genomics. There's very few high-quality genomes from humans relative to where the main focus has been, which is in Europe.
Yeah.
In modern Europeans. And so what's been well described, and I think we've even maybe hinted at it or talked about it a little bit on TWiEVO, is that there's been this integration event of Neanderthal DNA into European genomes, and that this still kind of echoes on between 1 or 3% of the genome might be attributed to an ancient hybridization event with Neanderthals. And then you have modern humans breeding for generations and generations, and there's just sort of a little remnant from some of those original hybridization events or matings between that archaic population, which is now extinct, and modern humans, which continue on today.
I think our paper we did was about a particular set of genes that we retain, right?
That's right. And then what people have done is gone forward, and we'll see this a little bit today, actually, and asked among the modern humans that retain that sort of remnant from the Neanderthal past, are there signs that some of that DNA, and especially in coding regions, has been held onto potentially for adaptive reasons? Mm-hmm. And so there's a proposal put forward based on some evidence that there's some immune encoding functions, the OAS cluster of genes, or oligoadenylate synthases in particular, that confer some protection against certain viruses, and that the genetic variant that you can find in some people might have been, in essence, donated or provided by that Neanderthal heritage.
Mm-hmm. Interesting. So it turns out that this area, well, first of all, this is Southeast Asia. And Papua, right? It's a collection of archipelagos bigger than Europe.
That's right. It's a pretty consequential,
huge.
Yeah.
And they say that the center of gravity of Denisovan introgression is the Papuan population, which I didn't realize.
I didn't either.
Pretty cool, right?
Very cool. And given, I think some of that geography and the islands and just the history of humans in that area is also really interesting, with a lot of, with what's the little that is known about genetic diversity in terms of genomic analysis, already pretty clear that there's a lot going on in that geographical location. And again, sort of underappreciated in terms of the level of analysis.
Yeah. Apparently there are few groups outside of this area with introgression from Denisovans. That's amazing, huh? Something went on there.
And something went on, and that's where this paper kind of picks up. And so there is this what's called, and has been running, I think, for more than a decade, the Indonesian Genome Diversity Project. And this is part of that effort. And for this sort of turn in that project, the authors are reporting on 161 individuals where they got 30x or greater than 30x coverage on the genomes. And I think this starts to reflect kind of this, exponential growth in genomic, or ability to sequence genomes at greater and greater depth. And then because of that, and given the possibilities of more and more comparisons, as you both get deeper and sort of go wider across human diversity, new sort of footholds for statistical analysis to push the inferences even farther, which is why I think this paper generated a lot of interest.
And these are people living now, right?
That's exactly right. And in fact, you can't, we don't know, they're not going to, obviously, for ethical reasons, identify the people, but they do provide, I think, a really useful map of the area in Figure 1A, which sort of illustrates across the 14 or so different geographical locations, the different islands, where they're doing this sampling of people who've lived there and have their heritage there. Their ancestors going back in these regions. And so already that map figure in 1A is more than just locations. It also gives an initial sense, or maybe like a higher level summary of in pie charts of what are the contributions to the genome. So both modern ancestry, so how much of the genome might be related to other sampling in East Asia versus more specific to Papuans.
Yeah.
And then also a first hint at the archaic ancestry, where they're trying to use those reference genomes, the 2 Neanderthals and the 1 Denisovan, to infer what might be introgressed. And so I think the take home here is that you actually see a pretty big contribution of the Denisovans the farther east you go into New Guinea, whereas the farther west you go into South Asia, it's actually Neanderthal and very little, if any, Denisovan contributions. And so that's sort of the first surprise, in a sense, is that this is and kind of gets at that really nice turn of phrase that you mentioned before, that center of gravity for thinking about Denisovan contributions.
Yeah, as you go north, also it goes way down. So the Denisovans were in a lot of places, but for some reason, a lot of introgression in the eastern part of this archipelago.
Yeah, exactly right. And I think, that it's a little bit of a cautionary note as well, because given the such small, we're still just at the infancy of archaic genomics. And, so you have that one sample from the Denisova cave in Siberia. And so obviously you might think that's the point of gravity, but as we were, or sort of the hotbed, but as we were describing, in terms of just the preservation of samples, being in a cold cave environment, and just the sort of limited amount that already we might sort of make these massive assumptions about the history of those people based on really scant details, or even sort of misleading information for where the real action might be happening.
Yeah.
So then, just as they're doing this analysis of these 160 individuals, they do a PCA, and I think that's just kind of a reality check or gut check. Are we sequencing folks that we think we're sequencing? So, for example, like if you moved to New Guinea last week and then somehow got into this project, that would probably be kind of a disaster for the to make inferences about the history in that area, since obviously your ancestors. I don't know actually if you've done your 23andMe ancestry, you're related, but I'm guessing you don't have a big.
No, we're mostly European. Yeah.
Yep. Same here. So anyway, that PCA, just sort of shared differences. You do start to see these groupings that make sense in terms of the folks that they're sampling from the different islands having a heritage that's kind of in common around those areas. I was kind of curious whether or not you could even put some of those archaic genomes into this PCA. I'm guessing there's so much divergence here that even with the Denisovans that might have been sort of in closer association, that the points might be way off chart, or would just kind of screw up the whole thing. So that's missing, but it's just the modern ones that and actually not just the ones they did, but taking a few, I think from West Eurasia, from Siberia, for example, and from the Americas, just to show a little bit of that genetic heritage. So kind of with that in mind, then the next question became to sort of take this broader spectrum, not only of, for comparison, not only of other human genomes, and I think they use about 300, so they roughly double the sampling pool that they're using for their statistical analysis. And these are samples, modern human samples from around the world. And then as we've been already kind of hinting at or discussing, using the 3 archaic genomes, the 2 Neanderthals and the Denisovan, to really get at this, and this gets back, I'd say even to the title or the take home of the paper, which is the deeply divergent Denisovan ancestries in Papuans. And so that will start to emerge from this. But they have that comparison point with the Neanderthals, which is sort of more of a European story in the history there. So anyway, to try to get at what are the contributions from these archaic human populations to the island Southeast Asia region, they really focus on what's called haplotype-based introgression analysis. And so we should probably maybe step back for a minute and do a couple of definitions. So first of all, introgression, we've already kind of hinted at this. This is this idea of hybridization. So the transfer of genetic material from one species to another, maybe put species in quotes, since these are obviously fertile hybrids when we're talking about our ancestors and then the archaic, now extinct human species or populations. And then to get at how, the further definition of introgression, this is followed by repeated backcrossing. And here backcrossing would just be Homo sapiens mating with Homo sapiens, moving forward from generation. So that's the introgression, the idea of haplotypes, which we've also talked about that's in some detail before, but this is just maybe as simply defined as a group of genetic variants inherited together from a single parent. So if we put this into sort of chromosomal context, you might have a genetic region that could span, a megabase or 100,000 base pairs or 10,000 base pairs that was the genetic contribution, that 50% contribution from one parent. And then the way that you would define that or kind of have a sense that it actually came from a single parent and not sort of both, or there was recombination or whatever, is that you have this group of variants that hitchhike or kind of ride together in a sense. And so it's the shared changes that define the haplotype from the other parent, sort of in this simplest case. So we're really talking about trying to use that sort of the connected nature of the chromosome in the changes across the DNA in these regions as a way of interpreting where the DNA originally came from, whether that's from one parent or the other, or whether that's from one species or another in this case. And then there's another sort of underlying definition that comes into this idea of incomplete lineage sorting, which is another sort of population genetics, sort of a major concept of understanding the relationships between individuals or how closely associated the genetics are between different groups. And so this definition is the idea of so if you're building a tree to map or to visualize phylogenetic relationships, that if you're just using a single gene, you might get fooled about how the population or the species are actually related to each other if you've kind of picked an outlier of variation in that gene. This happens quite a bit, actually. So, in part of the work that we do in my lab, thinking about changes, rapid evolution in genes encoding immune functions, for example, the evolution can be so rapid that if you build a tree taking that single gene, that immune function from different species, and then you compare that to sort of the accepted or the currently considered primate relationships between different species, say the human to gorilla to orangutan, etc. Sometimes the trees get a little bit scrambled because that single gene you've picked doesn't represent the entire population or the relationships. So that actually becomes a useful tool. Both something to keep an eye on, but also a useful tool for thinking about what are the changes and how basically these things can associate together. Okay, so anyway, a lot of definitions. What have they actually done here? So what they did is they hoped this notion of haplotype based introgression analysis. They aimed to find the largest. So as you're taking all of these genome sequences, you're trying to define large and specific haplotype blocks. And in fact, instead of going to the modern humans, they start with the Denisovan genome, that reference genome from that bone that was picked out of that Siberian cave. And then from there, they compare that against sub-Saharan African genomes to say, okay, how can I say this sort of fragment or haplotype of shared variation across a genetic region, which ones are the most Denisovan looking, meaning that they don't look like any of that great amount of diversity that you see in sub-Saharan Africa, which came later. And so that gives you that population differentiation or that species differentiation in certain regions of the genome. Then they continue to do that. They described using some statistical approaches, and maybe I'll just concede up front here. I'm not going to, I'm going to punt on some of the technical details of that. Some of the population genetics is a little beyond my pay grade, so to speak. So we may not get, a hypercritical analysis of that, but we'll kind of do our best to take what the authors are doing here and digest it to some degree. So they use several statistical approaches to filter the blocks by comparing them not only to the sub-Saharan Africans to say which are the most Denisovan, but they also compare that to the two Neanderthals, the other two reference genomes of archaic humans. And so from there, they have the quote unquote most Denisovan, remnants or parts of the genome. And they compare that now finally to their 161 samples, which kind of range across the island, Southeast Asia. And what they see is among about 32.3 megabases of their high confidence blocks per genome, that the Papuans really fall out, the easternmost samples, as looking sort of the most similar to the Denisovan haplotypes that they were able to define against the sub-Saharan Africans.
Yeah, it's not a lot, but it's there.
Yeah, so 32 megabases in 3 gigabases, right?
Yeah,
3 billion. So, and to say, there are also, that kind of fraction of the genome that you're considering, those blocks also aren't sort of homogeneous in size. So there are small blocks and there are larger blocks. And that will come back into play here as the analysis continues as well.
And they say in West Eurasians, there are only 688 kilobases.
Yeah, isn't that interesting? So there might be a kind of a little trace.
Yeah, very little.
Of that, but pretty modest. And I think there you also have to consider that these are kind of statistical definitions, right? So these aren't sort of one-to-one comparisons. And you're sort of necessarily really simplifying what is a massively complex sort of history of ancestry of mixing of humans, different populations in different ways as humans were migrating more and more. And so what you might define as Denisovan actually it's a more complicated thing than that. And so pulling signal from noise is not sort of a trivial exercise. But so that kind of with that foothold that they're proposing from that 32.3 megabases, they now go in and just focus on that part of the genome to ask even more about, and kind of with that focus on the Papuans as a subset of those 160 samples. What are the specifics of those Denisovan contributions? And so here we've kind of skipped ahead to figure 3. And I think it's really fascinating and well presented, I would say. So they're doing what they're calling mismatch distribution analysis in the haplotype blocks. And so what this reflects is that, obviously among these haplotypes, you can use the shared variants across the haplotype to define the haplotype. And yet, because there's been so much divergence, there are going to be mismatches. And so that gets kind of at that signal to noise zone. And so they're looking at the distribution of mismatches with sort of the simple idea that the more mismatches you have between the Denisovan haplotypes to the Papuan modern samples, that those are going to be potentially contributions from a non-reference strain. So who are these populations? So you only have one comparison. And so just by, it might be kind of it's not quite a circular argument, but just by analyzing the mismatches and sort of whether there's structure in the mismatches, meaning there's patterns that repeatedly come out, that they try to use as a signal to get at more specifics about what are these contributions and what does this mean for kind of Denisovan, to infer about that diversity as well. And so if you look at figure 3A, there's sort of an interesting heat map where on the x-axis, it's this measurement of mismatch distribution, and they just give that sort of a simple quantitative value. So if you're going from left to right, the more mismatched you are, the modern human from the Denisovan reference in that area of the genome. And then on the y-axis, it's the chunk, minimum chunk length. So that's literally the amount of the size of the haplotype in base pairs or kilobase pairs. And so what they see is they actually get a pretty good signal of differentiation from their samples as they go to the bigger chunks. And you see these, the heat map goes red. So they don't have as many of these large chunks of the genome, but there's what's interesting about the abundance of mismatches is that there are two hotspots, one at about 0.15 in the mismatch metric and one at about 0.2. And that actually is a kind of like two peaks. Mm-hmm. And what it suggests is that there's the introgression or the donation of the haplotype is from two different sources. One that's about 15, this is a little bit oversimplified, but one is about 15% different as a collection, and another that's about 20% different in a collection. And so if it was just a single population that was introgressing or donating in, all of the mismatches, making some assumptions, would just be the same amount of mismatch.
Right.
But here there's two unique sort of distributions of mismatches. And so they name these D1 and D2. And, it's a statistical definition, but the implication is that there's Denisovan population, ancient population 1 and ancient population 2. And because the D2 is a higher mismatch number, you would infer that introgression event happened earlier in time, or that it's been that was a more ancient genetic contribution to the Papuan individuals of today.
They say these two were separated for a very long time. What is that? Do you have any sense?
Yeah, so we'll get to that. That's sort of the next is dating. Not only when the introgressions might have happened, but actually when the Denisovan ancient populations might have diverged from the reference that's known. But before we get to there, just a quick note on a third population called D0. And that, if you look at figure 3C, so here again, it's the mismatch distribution and then the density. So how many of the haplotype blocks, the large ones in particular, that fall into that category of number of mismatches in the haplotypes. There's a little shoulder on the left, a little spike at about 0.05. This is much closer to and these are all the mismatches relate to the reference Denisovan. It's much closer to the reference. And so D0 would basically be a really close relative to the reference genome that was pulled out of that Siberian cave. And so there you see, that peak is the greatest in Siberia, not too surprising, East Asia, and that smaller contribution there. What's interesting is you also see a little bit of that in the Papua, but then those two peaks at a much higher density, which gets at sort of that this is the center of gravity or that related term for where these introgressions were happening.
So this means that they have sequences from at least two different Denisovan populations, right?
That's the idea. Exactly right. Yeah.
And so this obviously is having offspring with a Denisovan, right? Would that be a Homo sapiens who did that, or?
Yeah, right? Exactly right. Yeah, so that would be our ancestors.
Yeah.
In our lineage leading to today, and then and we'll talk about the dating in a minute here, but then the back crossing happening not as hybridizing. So there's going to be and I'd say I'm kind of oversimplifying. So we're talking in populations. We're not talking individual events. We're talking over windows of time, but at least from an evolutionary standpoint, as these sort of pivotal events that kind of echo on in the genetics of today. But yeah, that's right. So that's the idea, is that there are these population level contributions, but then that gets sort of whittled away as that window of hybridization closes, as intermating stop, as the archaic populations declined and eventually went extinct. And then who's left? It's sort of our close relatives and the modern humans of today.
So there could have been separate hybridizations, and likely were, because these are distant in time. And then the offspring with the introgression, they could have mated, and that brings the two different Denisovan sequences into one Papuan population, right?
Yep, that's right.
Okay.
And also, keep in mind all of the things that probably happened that we don't see today, that didn't survive. And that's kind of, evolution is always the things you can't see. And it's sort of the pin of the needle are the things that still exist, tip of the needle.
Now, they also apply this to Neanderthal sequences, the blocks, right?
Yeah, that's right.
And I don't see this bimodal distribution of mismatch.
Yeah, exactly. And in fact, there's very little relative to the signals that they're getting. So the density, meaning the number of haplotypes, right, that are fitting into these categories, it's a much more subtle signature for the Neanderthals if it's there at all. Whereas if this analysis were being done and has been done in European populations, it's the Neanderthal that are contributing to that modern heritage. Very little, if any, I think almost none, Denisovan in some of those cases. So it's kind of probably, obviously the geography mattered for where these matings were happening.
Yeah. Denisovans preferred this area. It's a little warmer.
That's right.
I can't blame them after living in that cave in Siberia for so long.
Yeah, I hear you. I mean, I also, I migrate south for the winter. Spring break kind of a scenario, perhaps, right? I mean, so I don't know enough about it, but there are, other peaks. I don't know if you noticed like that east, ISEA. So this is the islands of Southeast Asia. That's the purple part of the line going through. So there, instead of D1, D2, you have one
in between
peak kind of in between. And so that likely represents, I don't know how good the resolution is on this. Is that a third or a fourth population, a fourth peak, or does that represent one contribution from what would be considered D1 or D2 because they're kind of overlapping?
Yeah.
And so the kind of, the grade that's also, I think, a tricky part of this kind of analysis is that it's super impressive to see two clean signals, but then, what does one signal mean in terms of the resolution and kind of the gradient of geography, and sort of the messiness and complexity of mixing of a lot of ancient humans' genomes coming together in different ways.
Right.
Okay, so then finally, we're getting to your question, Vincent, which I think is a really good one, which is, so how do you start to think about like the date of this, or when did these things happen? And again, you can draw from a growing and I'd say increasingly powerful set of population genetics tools to start to put this into kind of a phylogenetic tree where you're trying to put dates on the branches closer to the trunk, working backwards in time. And so now, this is kind of another major concept in population genetics, which is coalescent modeling or coalescent theory. I think we've talked some about this too. It's basically how you look at gene variants that are sampled in a population today and consider that they were originated from a common ancestor where they weren't divergent, where they'd coalesce on the same. And so you are looking backwards, and then from modern sampling back to take into account a lot of variables, things like population sizes, things like mutation rates, time and correlated with time. Sometimes that's the molecular clock is connected to the fossil record. And then you use all of this to try to calculate a time to coalescence, or when was that last common ancestor before the genetic divergence into variants happened. And so they use this analysis with their data to do a couple of things. So the first is to look as deeply as they can. So this is back, they estimate something like 350,000 or 360,000 years ago, to ask when that D2 population, that peak that they inferred with that sort of shared mismatch distribution around 0.2, when do they think that population diverged from the others? And that's that's what they're estimating at 360,000 years. I would take this with a little grain of salt. So the confidence, 95% confidence intervals on that is about 50,000 years. And there are in the coalescent kind of related stuff, the molecular clock kind of stuff, even of today, with a lot of, pretty sophisticated genetic or statistical approaches, still really tricky. Lots of assumptions in those variables in terms of clock, the reliability of the clock, maybe haplotype specific assumptions, where not all of the genome has the same mutation spectrum and related things. And so anyway, they do put that dating on that. And then from there, the next thing they do is the divergence of that D1 spike, or that D1 peak, inferred population that's at about 0.15 mismatch on the large chunk haplotype analysis. And that they put at about 280,000 years ago. So this is, going from that one, that Siberian cave, Denisovan, and that was that's a much more modern, that's a much more modern ancient human that's only something like, I forget, maybe 40,000 years plus or minus. And so this brings the heritage of the Denisovans back quite a ways. And there's been other analysis in East Asian populations for that D0, that left peak, where it's much closer to the reference. And there they don't actually do the analysis. It's pretty heavy lifting from a statistical standpoint. But that is probably coming in something more like 100,000 years ago, and sort of the closest known link from integration analysis. So now that they have that kind of big, that's sort of like the Denisovan evolutionary tree, then they can actually, I think it's a really nicely illustrated figure. This is panel B, where they're now putting that Denisovan divergence then on top of modern humans in the diversification there, for when the introgression events happened. And so they're working from left to right. You've got the out of Africa event for modern humans dated to about 65,000 years ago. The idea of in human migrations that as you went up towards Europe, and then others taking a right turn into Asia, that you have the Papuan split at about 50,000 years ago. And then using their mismatch and their haplotypes, that's not too long after that split at about 45,000 years ago. They're inferring the D2 population introgressing into what is now modern Papuans. And then that D1 event, major event or major introgression happening at 30,000 years ago. So two events separated by about 15,000 years in time of intermingling between genetically unique archaic Denisovans.
So the D2 and the D1 Denisovans, did they not overlap temporally?
Well,
or they were just reproductively isolated?
So I might be wrong, but my impression is that's not known. And remember that the D2 and D1, it's a statistical definition based on differences in haplotype that are inferred from a small chunks of modern humans. So it's a great question. And the authors do at least say, I don't think they go that far, and I don't know that they can. But what they do say is that because of these dates, if they hold any water at all, what it does say is the Denisovans actually, the interesting thing is how recent actually those events have been, including that D0, and how ancient some of them are. So that sort of broad window of introgression, and that's, I think, pretty unique. I think the Neanderthals, it's thought that these were more sort of transient kind of windows of hybridization, and then which is now seen as introgression. But the Denisovans, it might have been a longer history of intermingling with modern humans, especially in the area in and around New Guinea. Yeah,
it seems that the Denisovans were around a long time, right?
Exactly. That's right. Yep.
And the way I read it, they say our model implies substantial reproductive separation of multiple Denisovan-like populations over a period of hundreds of thousands of years. So it sounds like they're coexisting, but they're separated. And somehow both of them get into the Papuan region to hybridize with the Homo sapiens there.
That's right. But so how much of that D1, D2, which is the signal in the analysis, versus how much is there intermingling between those? I think that is a pretty tough one to tackle.
So would it help to get more ancient Denisovan sequences, or because you're not going to get anything in between? Or maybe it would. Maybe if you find some specimens, right? But
no, I agree. That would be spectacular. I think that would be hugely informative to
get something and get something in between, not such an ancient one, right? Because the ancient, what's the dating on the cave Denisovan sequence? Do you know?
Yeah, I do. So, and that's well, I don't know, but I can see it in the figure. So if you look in 4B, that Altai Denisovan, Denny up to present, that's only something like 50,000 years ago. Oh. Very recent. Yep. And not to say, so actually the introgressions are dated to be even slightly more recent. You can kind of see how it aligns with that, the age of that sample, but the populations themselves, they're estimating to go back to reach back a long time. Yeah.
So you're not going to get much older than the Altai, I would guess, because they're not going to be preserved. Right.
That's right. So you're starting to get to the limit of being able to pull, robust DNA sequence from these archaic genomes. Really difficult. And that was something when we had Svante here out on the ski slopes, we were asking him, like, how far back do you think you can go? And if I'm remembering right, maybe 70,000. There's some horse fossils that are like 70,000 that people are getting some decent genomic data. But unless there's like some massive technological breakthrough, that might be starting to get to the ceiling. Yeah. So you can only,
what coverage do you get on these 50,000-year-old genomes?
Yeah.
Do you know?
Well, so it's getting better and better. I have to say that, and this was part of, obviously, Svante's, he did a public lecture here. And it was also kind of co-hosted by the Natural History Museum. Right. A lot of interest. So
sure.
it was kind of, it was kind of funny. So it was going to be, his talk was going to be at the Natural History Museum. It sold out in like 5 minutes. So they moved it to the football stadium.
Wow.
But that sounds a little bit, it's not, and by that, I don't mean like the 60,000 seats in the stadium, but there's a sort of luxury box area that can hold about 1,000 people. Maybe 900. And so, but then they told Svante that his talk was going to be in the football stadium. It's an outdoor stadium. This is in January. And, so I don't know what he was thinking. Like he was going to have like a football crowd in the middle of the winter. But anyway, it was a great event. Excuse me. It sold out, as well. So a lot of public interest. And so, he had a pretty, he gave a very kind of high level, public lecture, obviously. And so I didn't get a really good sense, and I have to confess, I haven't read, so I think a lot of this is published now, but I haven't read carefully of what are the kind of depths, that they're able to get these days, and actually how much coverage, which I think is even harder, because especially as you get into repetitive stuff in the genome, and contamination and everything else, those are the areas that sort of break down. And so when people say whole genome, even for modern species, the annotations can be a lot more modest. And so what does that really mean? It's always a good question to keep in mind. Okay. So then kind of the next turn for the paper was to use, to kind of correlate this idea of the Denisovans and all of this sort of new potential knowledge about the, both the diversity and the different populations stretching back in time, and to try to correlate that with some of the current thoughts about human migration history. This includes archaeological findings, other inferred introgressions. And again, I think it's important to keep that in mind, that context in mind of what the real single data point is, just that one bone from that cave in Denisovan. So again, maybe grain of salt kind of territory in some ways. And so, but using kind of connecting the geography and the idea that the eastern New Guinea area of the sort of island Southeast Asia, that this is where there are a lot of these collisions of ancient humans, that point that you kind of were hinting at, that the Denisovans survived a lot longer than Neanderthals and other. So and they, the authors point this out, and to kind of try to motivate more archaeological attention on Denisovans in general. I think the Pabo Lab, the folks up at Leipzig at the Max Planck Center there for Evolutionary Anthropology, are on the case. I think there's more samples kind of in the pipeline now, and I would expect that that's going to, be a really cool comparison point. So how good was the statistical analysis potentially for uncovering some of these patterns, or that they're proposing? Does real life in terms of the additional sampling sort of support that, or does it go in a new direction? I think that's still a fascinating and open question.
Yeah, for sure.
And I think, I would just step back too, and I think we've kind of been hinting at it, but to make the larger point too, just about the state of modern genomics. And this is mostly Europeans, and we're missing all of this super fascinating human history, consequential human history. And that matters not just for reconstructing our history, but it also matters for, as we're moving into sort of understanding genetic variation and the implications for things like medicine. There's massive blind spots currently, because we've barely kind of scratched the surface of even our own species in terms of the diversity there. Okay, so now kind of one or two, it's a pretty comprehensive paper. So one or two last turns, they wanted to ask, given the introgression that they've mapped, and by the way, I think for a lot of the Papuans, this could be like 4% of the genome might be accounted for by introgression from the Denisovans. Are there signs of that any of these are kind of being kept around because they're adaptive, that there's some benefit to the variants that that came from the Denisovans in those remaining, even despite the backcrossing, they remain perhaps for a beneficial reason. And so if you look at figure 6, they show a Manhattan plot, one of my favorite, of course, plots in genomics. So you just go on the x-axis, you're going chromosome to chromosome, and then measuring something that you're saying is interesting as you get peaks along the way. And it's sort of supposed to look like the Manhattan skyline as the skyscrapers are emerging from the smaller buildings. And so here, the y-axis is just the frequency. So among of that allele within the haplotype, or that variant within the haplotype that they're marking as Denisovan, and how many of those folks that they sampled have that Denisovan variant, might be a point mutation, et cetera, in those genes. And so if you just you can kind of, I kind of love looking at these, and just if I can, pull out any genes that my lab is studying or I've been interested in, and whether that kind of adds a cool connection to sort of ancient human history. But they do sort of a broader global analysis and ask among the, sets of genes, and it might be 30 or 50 genes that they're proposing. What are the categories? And one of the things that jumps to the forefront for enriched is immunity. Yeah. And so, right. Sure. And maybe not surprisingly,
you can see TNF and IL, interleukins, right?
Mm-hmm. Yeah, exactly. Exactly right. And they go on and do a little bit of a population genetic search for signs of positive selection, and have at least some hooks that way. And so that's consistent with this idea that some of these variants might have been, the adaptive nature of it would be that it is effective as an immune function, or it evades being inhibited by an infectious microbe in the past. And so that's kind of who lived, because they weren't crushed that infection. They went on to have kids. Yeah. They also see maybe a few genes in sort of potentially dietary adaptation that might be specific, but that's, I think, it's a pretty modest signal that's there.
So are any of these genes ones that you work on or have looked at, Nels?
Yeah, a little bit. So like some of the zinc finger, nothing that like just smacked me on the face and said, drop everything. And, but I might,
because the thing would be, if you had an antiviral protein, you could then reconstruct it and put it in culture and see what it does to contemporary viruses, right?
Yeah, exactly right. So we did, the closest we've come is not in Denisovans, but the Neanderthal, one of the variants that's been claimed to be sort of from the Neanderthal heritage in the Europeans in OAS1. Mm-hmm. We've taken that Neanderthal variant, put that into a yeast assay that allows us to test the activity of the gene. And I think the activity was a little bit higher. That gets back to that TWiEVO we did from Luis Barrero's lab. And we actually saw something like that when we did our, we were actually, and a lot of the studies we do are more kind of primate focused. So we go back, instead of going back this 50,000 or 60,000 years, we generally go back 35 million years to sample that net of diversity and see what are the other primates up to in terms of the genetic diversity among their immune functions. And so that was the main thrust. This is a paper we published just a month or two ago in Cell Host Microbe. Right.
I think I had suggested we do that, but you didn't want to do it.
No, not true. You just caught me at we're in that academic sprint right now, right? And the teaching, everything else. And so unfortunately, the timing wasn't right. But don't worry, we have plenty more interesting stories in the
for sure.
And you know where you know how to find me on the podcasting frequency. So we'll be back. Okay, so then maybe just to kind of conclude, but they finally they step back. So up until now, they've really been kind of focused just on a subset of their data, that 32 megabases or so of the haplotypes that they're kind of high confidence in terms of both size and kind of identity back to the reference Denisovan. So they actually went back and did sort of to the original haplotype analysis, and this kind of considers other potential signatures or signals of integration from other archaic populations. And so maybe the most obvious candidate for this geographical region would be Homo floresiensis. Yep. And then they even say that they kind of looked for Homo erectus, and now we're back, a million years. And I think the outcome there was, and so they're looking, for minimal overlap in the mismatches.
Yeah.
But that's pretty tough. I think basically they're kind of getting maybe in terms of this analysis, the signal to noise starts to catch up with them pretty quickly.
Yeah. Sure.
As you really try to go back. And so they don't see, and so I would say the jury's still out. Maybe there are contributions. It's just really hard to see the just faint traces of them. Or maybe they're not. It's still, that's kind of, I think, left as a loose thread.
They mentioned specifically this village where they had found others had found floresiensis bones. Right. So you would guess, well, this is a good place to look for introgression, right?
Yeah, that's right. So the archaeological link would make, then you could ask, and I think the genetics statistics can't quite, and that, again, who knows, that could be there, but it's just not. And at some point too, I guess, you might also consider what are the hybridization limits as well. And so maybe those were sort of the failed experiments in human sort of reproductive history in a sense, or evolutionary history. So that's about it. And, pretty cool, I would say, to really go from that kind of single reference Denisovan to kind of thinking about, by just looking kind of in the right place, to start to uncover these pretty intriguing signals of diversity coming from
two new Denisovan groups that they show bred with modern humans. And
at least by statistics.
And as you said, about 4% of the Papuan genome seems to have been contributed by this.
Yeah, kind of a who's who of your genome.
Yeah. But I'm struck with the larger contribution in this area than elsewhere. So that's very interesting. I put a little Denisovan map in the show notes. It's a migratory map, I think. Yes.
Yeah, that's cool.
So showing them, there's got the cave there. Then there's a line heading over to the Middle East. And then there's a line heading down to Southeast Asia where we've been doing this study today. And that's it as far as we know. It didn't go into Africa. They didn't go into Europe. The Denisovans, they didn't go into the Western Hemisphere at all.
Yeah, no, I don't. I think that's the current state of affairs that they, and actually, I think the authors hint at it. So just from the archaeology, it's pretty impressive how much ground they covered.
Yeah.
Given sort of that time in human history, crossing the Wallace line, which is this sort of famous naturalist definition, Alfred Russel Wallace,
right.
who identified in island Southeast Asia the difference in animals and being pretty, that the set of animals like the marsupials, et cetera, that you see in Australia versus the ones that you see in Asia as a pretty for outside of humans being a pretty massive divide in animal diversity. The Denisovans went right across.
They could cross these barriers. That's pretty interesting. Maybe they built little boats,
Yep. And this obviously was a much later migration than as the continents were pulling apart and animals were diversifying and were separated out that way.
Modern humans experienced repeated and persistent interactions as they expanded out of Africa into this highly structured archaic landscape. This genetic contact yielded a rich legacy. So we're not just Homo sapiens, man.
Yeah, that's right. We've got some traces.
As you would say, little spices of other archaic humans, Denisovans, Neanderthals, and who knows what else.
That's right. And stay tuned, because I think, more on the way. And as the techniques get more powerful, as the genomics goes deeper, and also expect some revisions, I think, as well. Yep.
I mean, this is based on the that one finger sequence, and, it might all be out the window if there's something wrong with that, right?
Yeah, yes, that's right. I think there's more to be said about archaic sequencing, but it will only, the confidence, I think, gets higher the farther we go. And imagine it was only, I don't know, maybe 15 years ago when archaic genomics meant sequencing your own mitochondrial DNA and reporting a complete contaminated sample of yourself.
Yeah, right.
And so it's a pretty incredible trajectory in terms of the field moving forward.
But, and tell me if this is the right way to think about it. So, we have a few percent of our genome is Neanderthal. And in some parts of the world, a few percent is both Neanderthal and Denisovan. The rest is chimp, basically, right?
Well, human, yeah.
It's human. It's moved away, but it's very,
sure.
It's 98% similar to chimp DNA, right?
Yeah, that's right. And so that kind of reflects a different measure, which is, so there we're mostly, when we're thinking about the deeper connection of gene conservation, usually we're talking about coding sequences.
Yeah.
And there we're talking about functions that have been conserved. So like the actin gene, where you can see clear identity all the way out to yeast in a common ancestor that's back, hundreds and hundreds of millions of years. And so, the haplotypes, very different animal, because we're nothing about any kind of conservation. It's just genetic variation. And so, so take that chimp human number with a grain of salt, or put it in that context of being the conserved gene number.
So it's not the entire exome then?
That's probably the entire exome. But even that, some are 100% identical.
Yeah.
And others are less, if you go to the immunity genes, for example. And so it can be a pretty broad range.
So if you compared a Denisovan with a chimp, you'd get a similar number, I guess, right?
Correct. That's right. Exactly right.
Yep. Okay. Cool paper, Nels. Very good.
Yeah, that was fun. Yeah, I enjoyed it. Should we hit the old mailbag?
Yep. We got 2 here we can do.
Okay.
Take that first one.
I will. So Scott writes, "Hi, Nels and Vincent. Firstly, thank you for an excellent and fascinating podcast. I found you via TWiEVO, and I've been listening for a couple of years now. My favorite episode was TWiEVO 24 with Marco Vignuzzi. I think I listened to it at least 4 times to try to get an understanding of the quasi-species concept as it relates to RNA viruses. I also enjoyed the most recent episode about antifreeze proteins in cod. I work in a lab that develops vaccines for farmed fish, and anything fish-related always gets my attention. On that note, I recently came across this fascinating research from Peter Unmack and colleagues on the persistence of the so-called ghost genomes in a population of fish in Australia. These are parasitic haploid genomes of an effectively extinct species that persist in a living population of hybrids. It might even make a nice paper for discussion on the podcast. Links to the original article and a nice blog post by the authors is copied below. Keep up the great work, Scott. P.S. It's a beautiful, sunny autumn morning here in Tasmania with a temperature of 13.5 C, but the first frosts are probably not far away." Hey, thanks, Scott. Really nice message. I'm a big fan of that TWiEVO 24 as well, having Marco in studio. A really fun conversation. Really interesting, incredibly gifted scientist doing great stuff. And I agree, that quasi-species concept is a tough one. And, kind of gets at this idea that are there kind of complex sort of genetic contributions at a population level that coming together are potentially adaptive for that virus. That's still a pretty controversial idea. It's still an active area of research. Another fellow who's now in Australia, Eddie Holmes, is a pretty strong voice in sort of throwing a little note of caution on the quasi-species concept. And so it's worth kind of following Eddie and some of, I think, of his points on thinking about RNA virus evolution. I think this ghost genome paper sounds really interesting. We'll take a deeper look at this. And fun echoes here, Papua New Guinea, Australia, Tasmania, all in this part of the world with some great and incredible genetic diversity to consider.
Hemiclonal gudgeons, for those who thought sex was simple. Interesting. Yeah, let's take a look at this. Maybe we'll do it.
Yeah.
All right, we have one from Ornob. Hello, Nels and Vincent. This is Ornob from Bangladesh. I had written to you guys earlier asking for advice about PhD applications and would like to first thank you for your informative and thoughtful answers. I have since applied and gotten into a few programs and will be starting at NYU this fall. Programs and professors at NYU offer a lot in terms of learning computational skills in population genetics, and I am super excited. Hey, congratulations, man. Very good.
Yeah, that's awesome. Great place to do PhD training. And I'd say, Vincent, let's pencil in Ornob for future TWiEVO, depending on the direction his PhD research goes.
For sure. Yeah, one of many here in New York City, right?
Yeah, that's right.
Continuing with his letter. Second, I was really excited to come across this new paper on the evolution of the RAG recombinase and thought it might be cool for a TWiEVO episode. You've probably already seen it, but the paper is called Transposon Molecular Domestication and the Evolution of the RAG Recombinase, and here is a link. It's a Nature article. Yep. Another one we could look at, right?
Totally agree. I did see that one, kind of on the same page here, or now I agree. Really interesting. I was thinking about proposing that for today's episode. Maybe this could be a new hybrid podcast of immune and TWiEVO. ImmuneEvo. I don't know. I'll have to work on the hybridization of the title, but yeah, very interesting.
This is of course, a RAG recombinase involved in VDJ recombination, right?
That's right. Somatic hypermutation to kind of the basis of generating antibody diversity.
And that's in antibodies and T cell receptors. So this looks at the evolution of that, right?
Yeah, that's right. Which is increasing. I mean, so the idea of a transposon heritage, that idea and some evidence for that been along for a while, but I think that's really some fundamental steps forward and really putting getting a grasp on how that might have happened from an evolutionary standpoint. It's a good time to revisit.
Yeah, maybe, I don't know, Nels, maybe you have to come on Immune and we could do that over there.
Yeah, that'd be fun. Count me in.
And figure out how to hybridize that title. Yep.
I'll work on that one in the meantime.
All right. Thanks, guys, for your letters. If you want to send a letter, it's TWiEVO at microbe.tv. We'd love to hear from you.
And just to say, really appreciate the paper recommendations. That definitely played a role in picking today's paper. So even if we don't get to them right away or get to them at all, we really love hearing back on ideas.
You bet. How about some picks,
okay,
Nels?
Science picks of the week. So I think as many of our listeners know, I'm sure as you know, Sydney Brenner passed away a few weeks ago, I think, or first week of April. And so in memory of Sydney Brenner, who I think was in his early 90s, maybe 92, I have a link to a talk or conversation he gave. It's on the iBiology YouTube channel entitled What Genomes Can Tell Us About the Past. And just kind of a fun glimpse. I think it was recorded maybe about 10 years ago when he was in his early 80s. But to just hear him talk about some of the really foundational concepts in comparative genetics, really a giant in the field of molecular biology, obviously with his shared Nobel Prize for genetic code and related stuff. And then I think really uniquely in some ways, just great fundamental contributions again and again throughout his career. Really impressive. And so that's my science pick of the week is that. Yep.
It's funny. They put him in front of a green screen and then they didn't use it.
Yeah, they didn't actually
didn't put anything on it. Didn't substitute anything.
Yeah. So, and I don't know how maybe I'm wrong, but I thought iBiology was more recent. But yeah, it's definitely, it was recorded 10 years ago, and it's not the most slick presentation, but it's just, it has a little bit of that energy of just kind of hanging out for a drink with your uncle at a bar or something like that.
Yeah. Well, maybe it's just a thing they recorded while he was there doing something else. Who knows?
Yeah.
Right. He recorded a formal lecture, and then they said, let's talk about this or something. Yeah.
Yeah, it could be. Yep. How about you, Vincent? What's your science pick of the week?
Well, I have an article in PNAS, which is not about experimental science, but it's about science communication. It's called Scientific Communication in a Post-Truth Society. And this is from Shanto Iyengar and Doug Massey. I don't know if it's open access or not. Let's see if I can tell.
Oh, yeah, good question.
Usually it says. But
right. I'm not seeing it.
It's a really interesting, for me particularly, because I'm interested in science communication, and this is all about the history of communicating and why it's led to the present state, where our internet is full of misleading and biased information. And they go through the history. We explain how these shifts, the profound shifts in media environment that have occurred, have enabled unscrupulous actors with ulterior motives to circulate fake news, misinformation, and disinformation. We document the high degree of partisan animosity, implicit ideological bias, political polarization, and politically motivated reasoning that now prevail in the public sphere. And then they say, what can we do about this? It's quite an interesting history. I really think it's important to take a look at this. And
yeah, I agree.
I just, let me see, I'll pull down the PDF. Usually that says whether it's open access or not. Let's see, it's a direct submission. Doesn't look like open access, unfortunately. I'm very sorry about that.
Oh, yeah. Well, a little bit of irony there. But you can always, so in cases like this, if you don't have easy access to PNAS or other paywalled journals, you can always write to the authors.
Yeah.
Yep. And they can immediately shoot you a PDF. And most are really happy to do that.
Yeah.
And yep.
And this, we'll put a link in. That'll get you the abstract so you can get the author's email, and then you could send it to them. It's cool.
Yep.
All right. That is TWiEVO number 42. You can listen on your phone or tablet. You use a podcast player. Please subscribe to TWiEVO so we know how many people are out there. I'm always surprised by who writes, and we'd love to hear more. So please subscribe, and of course, and that's free. And if you really like what we do, consider supporting us. You can go to microbe.tv/contribute for a number of ways, including, give $1 a month. That would help us a lot. And for that, you get all the podcasts of Microbe TV, 4 TWiVs a month, 2 TWIPs, 2 Microbe TVs, 1 TWiEVO, and 1 Immune. It's a great deal for $1 a month, don't you think, Nels?
I agree, 100%.
And of course, questions and comments go to TWiEVO at microbe.tv. We are dinosaurs here, Nels and I. We still use email.
That's right.
But it works. It still works. Nels Elde's at cellvolution.org, and over on Twitter, he's L Earlybird. Thanks, Nels.
Yeah, thank you, Vincent. Sorry we didn't do it in vivo when I was in your neck of the woods, but this is fun as usual, and can't wait for the next.
We'll have a couple this summer. We'll try and get together.
Yeah.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiEVO is by Trampled by Turtles. You can hear him at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.